Especially in the early years, there is a great deal of plasticity in the organization of the nervous system as patterns of connection between neurons develop. For example, if a child is born with its eyes misaligned, a condition known as strabismus, he or she will use mostly one eye. The connections to the brain from that eye will become strong, and connections from the other eye will become weak. The child will develop with reduced visual acuity and depth perception. If alignment is corrected in the first 3 years of life, however, the connections between the eyes and the brain will correct themselves, and the child will develop normal vision. If the eye alignment is corrected after 3 years of age, the correct connections between the eyes and the brain will not develop, and the visual impairment will be irreversible. Thus, plasticity in the development of the visual system in humans continues for several years after birth. A very exciting area of current research is the role of learning in stimulating the production and differentiation of new neurons in the brains of young and even adult animals. Institute for Advanced Studies, Jodhpur 33. C1. Cell aggregation and differentiation in dictyostelium. Differentiation and morphogenesis in dictyostelium, cell adhesion. The life cycle of dictyostelium. Another type of multicellular organization derived from unicellular organisms is found in Dictyostelium discoideum. The life cycle of this fascinating organism is illustrated in figure. Cycle. Solitary haploid amoebae, called myxomobae or social amoebae, to distinguish them from amoeba species that always remain solitary, live on decaying logs, eating bacteria and reproducing by binary fission. When they have exhausted their food supply, tens of thousands of these myxomobae join together to form moving streams of cells that converge at a central point. Here they pile atop one another to produce a conical mound called a tight aggregate. Subsequently, a tip arises at the top of this mound, and the tight aggregate bends over to produce a migrating slug with the tip at the front. The slug, often given the more dignified title of Pseudoplasmodium or grex, is usually 2 to 4 long and is encased in a slimy sheath. The grex begins to migrate if the environment is dark and moist, with its anterior tip slightly raised. When it reaches an illuminated area, migration ceases, and the grex differentiates into a fruiting body composed of spore cells and a stalk. The anterior cells, representing 15 to 20% of the entire cellular population, form the tube stalk. This process begins as some of the central anterior cells, the prestalk cells, begin secreting an extracellular coat and extending a tube through the grex. As prestalk cells differentiate, they form vacuoles and enlarge, lifting up the mass of prespore cells that had made up the posterior 4/5 of the grex. The stalk cells die, but the prespore cells, elevated above the stalk, become spore cells. These spore cells disperse, each one becoming a new myxamoeba. In addition to this asexual cycle, there is a possibility for sex in dictyostelium. 2 myxamobae can fuse to create a giant cell, which digests all the other cells of the aggregate. It has eaten all its neighbors, it insists itself in a thick wall and undergoes meiotic and meiotic divisions. Eventually, new myxamobae are liberated. Dictyostelium has been a wonderful experimental organism for developmental biologists because initially identical cells are differentiated into one of 2 alternative cell types, spore and stalk. It is an organism wherein individual cells come together to form a cohesive structure composed of differentiated cell types, akin to tissue formation in more complex organisms. Aggregation of thousands of myxamobae into a single organism is an incredible feat of organization that invites experimentation to answer questions about the mechanisms involved. Aggregation is initiated as each of the cells begins to synthesize CAM. There are no dominant cells that begin the secretion or control the others. Rather, the sites of aggregation are determined by the distribution of myxamobae. Neighboring cells respond to CAMP in 2 ways. They initiate a movement toward the CAMP pulse, and they release CAMP of their own. After this, the cell is unresponsive to further CAMP pulses for several minutes. The result is a rotating spiral wave of CAMP that is propagated throughout the population of cells. As each wave arrives, the cells take another step toward the center. The differentiation of individual myxamobae into either stalk, somatic, or spore reproductive cells is a complex matter. Rapert 1940 and Bonner 1957 demonstrated that the anterior cells normally become stalk, while the remaining, posterior cells are usually destined to form spores. However, surgically removing the anterior part of the slug does not abolish its ability to form a stalk. Rather, the cells that now find themselves at the anterior end, and which originally had been destined to produce spores, now form a stalk. Somehow a decision is made so that whichever cells are anterior become stalk cells and whichever are posterior become spores. The ability of cells to change their developmental fates according to their location within the whole organism and thereby compensate for missing parts is called regulation. We will see this phenomenon in many embryos, including those of mammals. Institute for Advanced Studies, Jodhpur 34. Cell adhesion molecules in dictyostelium. How do individual cells stick together to form a cohesive organism? This problem is the same one that embryonic cells face, and the solution that evolved in the protist is the same one used by embryos, developmentally regulated cell adhesion molecules. While growing mesotically on bacteria, dictyostelium cells do not adhere to one another. However, once cell division stops, the cells become increasingly adhesive, reaching a plateau of maximum cohesiveness around 8 hours after starvation. Cell adhesion is mediated by a 24,000 Dalton, 24 kDa glycoprotein that is absent in myxamobae but appears shortly after division ceases. This protein is synthesized from newly transcribed mRNA and becomes localized in the cell membranes of the myxamobae. If myxamobae are treated with antibodies that bind to and mask this protein, they will not stick to one another, and all subsequent development ceases. Once this initial aggregation has occurred, it is stabilized by a second cell adhesion molecule. This 80 kDa glycoprotein is also synthesized during the aggregation phase. If it is defective or absent in the cells, small slugs will form, and their fruiting bodies will be only about 1/3 the normal size. Thus, the second cell adhesion system seems to be needed for retaining a large enough number of cells to form large fruiting bodies. In addition, a third cell adhesion system is activated late in development, while the slug is migrating. This protein appears to be important in the movement of the prestalk cells to the apex of the mound. Thus, dictyostelium has evolved 3 developmentally regulated systems of cell adhesion that are necessary for the morphogenesis of individual cells into a coherent organism. Metazoan cells also use cell adhesion molecules to form the tissues and organs of the embryo. Dictyostelium is a part-time multicellular organism that does not form many cell types, and the more complex multicellular organisms do not form by the aggregation of formally independent cells. Nevertheless, many of the principles of development demonstrated by this simple organism also appear in embryos of more complex phyla. The ability of individual cells to sense a chemical gradient, as in the myxamoeba's response to CAM, is very important for cell migration and morphogenesis during animal development. The role of cell surface proteins in cell cohesiveness is seen throughout the animal kingdom, and differentiation-inducing molecules are beginning to be isolated in metazoan organisms. Differentiation in dictyostelium. Differentiation into stalk cell or spore cell reflects another major phenomenon of embryogenesis, the cell selection of a developmental pathway. Cells often select a particular developmental fate when alternatives are available. A particular cell in a vertebrate embryo, for instance, can become either an epidermal skin cell or a neuron. In Dictyostelium, we see a simple dichotomous decision, because only two cell types are possible. How is it that a given cell becomes a stalk cell or a spore cell? Although the details aren't fully known, a cell's fate appears to be regulated by certain diffusible molecules. The two major candidates are differentiation-inducing factor, DIF, and CAM. DIF appears to be necessary for stalk cell differentiation. This factor, like the sex-inducing factor of Volvox, is effective at very low concentrations 10 to 10 m and, like the Volvox protein, it appears to induce differentiation into a particular type of cell. When added to isolated myxomobae or even to press-port posterior cells, it causes them to form stalk cells. Synthesis of this low molecular weight lipid is genetically regulated, for there are mutant strains of Dictyostelium that form only spore precursors and no stalk cells. When DIF is added to these mutant cultures, stalk cells are able to differentiate, and new press-stalk-specific mRNAs are seen in the cell cytoplasm. Mechanisms by which DIF induces 20% of the Grex cells to become stalk tissue are still controversial. DIF may act by releasing calcium ions from intracellular compartments within the cell. Although DIF stimulates myxomobae to become press-stalk cells, the differentiation of press-pore cells is most likely controlled by the continuing pulses of CAM. High concentrations of CAMP initiate the expression of press-pore-specific mRNAs in aggregated myxomobae. Moreover, when slugs are placed in a medium containing an enzyme that destroys extracellular CAM, the press-pore cells lose their differentiated characteristics. Biochemistry of this reaction involves a receptor that binds CAM. When it occurs, specific gene transcription takes place, motility toward the source of the CAMP is initiated, and adenyl cyclase enzymes which synthesize CAM from ATP are activated. The newly formed CAMP activates the cell's own receptors, as well as those of its neighbors. The cells in the area remain insensitive to new waves of CAMP until the bound CAMP is removed from the receptors by another cell surface enzyme, phosphodiesterase. The mathematics of such oscillation reactions predict that the diffusion of CAMP should initially be circular. However, as CAMP interacts with the cells that receive and propagate the signal, the cells that receive the front part of the wave begin to migrate at a different rate than the cells behind them. The result is the rotating spiral of CAMP and migration. Interestingly, the same mathematical formulas predict the behavior of certain chemical reactions in the formation of new stars in rotating spiral galaxies. Institute for Advanced Studies, Jodhpur 35. Eye-lens induction. Organs are complex structures composed of numerous types of tissues. In the eye, for example, light is transmitted through the transparent corneal tissue and focused by the lens tissue, the diameter of which is controlled by muscle tissue, eventually impinging on the tissue of the neural retina. The precise arrangement of tissues in this organ cannot be disturbed without impairing its function. Cell ordination in the construction of organs is accomplished by one group of cells changing the behavior of an adjacent set of cells, thereby causing them to change their shape, meiotic rate, or fate. This kind of interaction at close range between two or more cells or tissues of different history and properties is called proximate interaction, or induction. There are at least two components to every inductive interaction. The first component is the inducer, the tissue that produces a signal or signals that changes the cellular behavior of the other tissue. The second component, the tissue being induced, is the responder. Not all tissues can respond to the signal being produced by the inducer. For instance, if the optic vesicle, presumptive retina, of Xenopus laevis is placed in an ectopic location, i.e., in a different place from where it normally forms underneath the head ectoderm, it will induce that ectoderm to form lens tissue. Only the optic vesicle appears to be able to do this, therefore, it is an inducer. However, if the optic vesicle is placed beneath ectoderm in the flank or abdomen of the same organism, that ectoderm will not be able to respond. Only the head ectoderm is competent to respond to the signals from the optic vesicle by producing a lens. This ability to respond to a specific inductive signal is called competence. Competence is not a passive state, but an actively acquired condition. For example, in the developing chick in the mammalian eye, the PAX6 protein appears to be important in making the ectoderm competent to respond to the inductive signal from the optic vesicle. PAX expression is seen in the head ectoderm, which can respond to the optic vesicle by forming lenses, and it is not seen in other regions of the surface ectoderm. Moreover, the importance of PAX6 as a competence factor was demonstrated by recombination experiments using embryonic rat eye tissue. The homozygous PAX6 mutant rat has a phenotype similar to the homozygous PAX6 mutant mouse, lacking eyes and nose. It has been shown that part of this phenotype is due to the failure of lens induction. But which is the defective component, the optic vesicle or the surface ectoderm? When head ectoderm from PAX6 mutant rat embryos was combined with a wild-type optic vesicle, no lenses were formed. However, when the head ectoderm from wild-type rat embryos was combined with a PAX6 mutant optic vesicle, lenses formed normally. Therefore, PAX6 is needed for the surface ectoderm to respond to the inductive signal from the optic vesicle. The inducing tissue does not need it. It is not known how PAX6 becomes expressed in the anterior ectoderm of the embryo, although it is thought that its expression is induced by the anterior regions of the neural plate. Competence to respond to the optic vesicle inducer can be conferred on ectodermal tissue by incubating it next to anterior neural plate tissue. Thus, there is no single inducer of the lens. Studies on amphibians suggest that the first inducers may be the pharyngeal endoderm and heart-forming mesoderm that underlie the lens-forming ectoderm during the early and mid-gastrulous stages. The neural plate may produce the next signals, including a signal that promotes the synthesis of PAX6 in the anterior ectoderm. Thus, the optic vesicle appears to be the inducer, but the anterior ectoderm has already been induced by at least two other factors. The situation is like that of the player who kicks the winning goal of a soccer match. Cascades of induction, reciprocal and sequential inductive events. Another feature of induction is the reciprocal nature of many inductive interactions. Once the lens has formed, it can then induce other tissues. One of these responding tissues is the optic vesicle itself. Now the inducer becomes the induced. Under the influence of factors secreted by the lens, the optic vesicle becomes the optic cup, and the wall of the optic cup differentiates into two layers, the pigmented retina and the neural retina. Such interactions are called reciprocal inductions. Institute for Advanced Studies, Jodhpur 36. At the same time, the lens is also inducing the ectoderm above it to become the cornea. Lens-forming ectoderm. The cornea-forming ectoderm has achieved a particular competence to respond to inductive signals, in this case the signals from the lens. Under the influence of the lens, the corneal ectodermal cells become columnar and secrete multiple layers of collagen. Mesenchymal cells from the neural crest use this collagen matrix to enter the area and secrete a set of proteins, including the enzyme hyaluronidase, that further differentiate the cornea. A third signal, the hormone thyroxine, dehydrates the tissue and makes it transparent. Thus, there are sequential inductive events, and multiple causes for each induction. C3. Limb development in vertebrates. Pattern formation is the process by which embryonic cells form ordered spatial arrangements of differentiated tissues. The ability to carry out this process is one of the most dramatic properties of developing organisms, and one that has provoked a sense of awe in scientists and laypeople alike. How is it that the embryo is able not only to generate all the different cell types of the body, but also to produce them in a way that forms functional tissues and organs? It is to differentiate the chondrocytes and osteocytes that synthesize the cartilage and bone matrices, respectively. It is another thing to produce those cells in a temporal spatial orientation that generates a functional bone. It is still another thing to make that bone a humerus and not a pelvis or a femur. The ability of limb cells to sense their relative positions and to differentiate with regard to those positions has been the subject of intense debate and experimentation. How are the cells that differentiate into the embryonic bone specified so as to form an appendage with digits at one end and a shoulder at the other? It would be quite a useless appendage if the order were reversed. Here the cell types are the same, but the patterns they form are different. The vertebrate limb is an extremely complex organ with an asymmetrical arrangement of parts. There are three major axes to consider, one of which is the proximal. Institute for Advanced Studies, Jodhpur 37. Close to distal far axis. The bones of the limb, wing, foot, hand, or flipper, consist of a proximal stylopod, humerus, femur, adjacent to the body wall, a zygopod, radius ulna, tibia, fibula in the middle region, and a distal autopod, carpals, fingers, tarsals, toes. Originally, these structures are cartilaginous, but eventually, most of the cartilage is replaced by bone. The positions of each of the bones and muscles in the limb are precisely organized. The second axis is the anterior, front to posterior, back axis. Our little fingers, for instance, mark the posterior side, while our thumbs are in the anterior. In humans, it is obvious that each hand develops as a mirror image of the other. One can imagine other arrangements to exist, such as the thumb developing on the left side of both hands, but these do not occur. The third axis is the dorsal ventral axis. The palm ventral is readily distinguishable from the knuckles dorsal. In some manner, the three-dimensional pattern of the forelimb is routinely produced. Fundamental problem of morphogenesis: how specific structures arise in particular places, is exemplified in limb development. How is it that one part of the lateral plate mesoderm develops limb-forming capacity? How is it that the fingers form at one end of the limb and nowhere else? How is it that the little finger develops at one edge of the limb and the thumb at the other? The basic, morphogenic rules for forming a limb appear to be the same in all tetrapods. 1977 showed that grafted pieces of reptile or mammalian limb buds can direct the formation of chick limbs, and Sessions and co-workers 1989 found that regions of frog limb buds can direct the patterning of salamander limbs, and vice versa. Moreover, the regeneration of salamander limbs appears to follow the same rules as developing a limb. But what are these morphogenic rules? The positional information needed to construct a limb has to function in a three-dimensional coordinate system. In the past decade, particular proteins have been identified that play a role in the formation of each of these limb axes. The proximal distal shoulder finger, hip-toe axis appears to be regulated by the fibroblast growth factor FGF family of proteins. The posterior thumb pinky axis seems to be regulated by the sonic hedgehog protein, and the dorsal ventral knuckle palm axis is regulated, at least in part, by WNT7A. The interactions of these proteins determine the differentiation of the cell types and also mutually support one another. Formation of the limb bud. Specification of the limb fields, Hox genes and retinoic acid. Limbs will not form just anywhere along the body. Rather, there are discrete positions where limb fields are generated. Using the techniques, researchers have precisely localized the limb fields of many vertebrate species. Interestingly, in all land vertebrates, there are only 4 limb buds per embryo, and they are always opposite each other with respect to the midline. Although the limbs of different vertebrates differ with respect to which somite level they arise from, their position is constant with respect to the level of Hox gene expression along the anterior-posterior axis. For instances in which the pectoral and pelvic fins correspond to the anterior and posterior limbs, respectively amphibians, birds, and mammals, the forelimb buds are found at the most anterior expression region of Hox6, the position of the first thoracic vertebra. The lateral plate mesoderm in the limb field is also special in that it will induce myoblasts to migrate out from the somites and enter the limb bud. No other region of the lateral plate mesoderm will do that. Retinoic acid appears to be critical for the initiation of limb bud outgrowth, since blocking the synthesis of retinoic acid with certain drugs prevents limb bud initiation. 1992 suggested that a gradient of retinoic acid along the anterior-posterior axis might activate certain homeotic genes in particular cells and thereby specify them to become included in the limb field. The source of this retinoic acid is probably Henson's node. Specification of limb fields by retinoic acid-activated Hox genes might explain a bizarre observation made by Mohani Hamidi and colleagues 1992 and repeated by Maiden 1993. When the tails of tadpoles were amputated and the stumps exposed to retinoic acid during the first days of regeneration, the tadpoles regenerated several legs from the tail stump. It appears that the retinoic acid caused a homeotic transformation in the regenerating tail by respecifying the tail tissue as a limb-forming pelvic region. Induction of the early limb bud, fibroblast growth factors. Development begins when mesenchyme cells proliferate from a somatic layer of the limb field lateral plate mesoderm, limb skeletal precursors, and from the somites limb muscle precursors. These cells accumulate under the epidermal tissue to create a circular bulge called a limb bud. Recent studies on the earliest stages of limb formation have shown that the signal for limb bud formation comes from the lateral plate mesoderm cells that will become the prospective limb mesenchyme. Institute for Advanced Studies, Jodhpur 38. These cells secrete the paracrine factor FGF10. G is capable of initiating the limb-forming interactions between the ectoderm and the mesoderm. If beads containing FGF10 are placed ectopically beneath the flank ectoderm, extra limbs emerge. Specification of forelimb or hind limb, TBX4 and TBX5. The limb buds have to be specified as being those of either the forelimb or the hind limb. How are these distinguished? In 1996, Gibson, Brown, and colleagues made a tantalizing correlation. The gene encoding the TBX5 transcription factor is transcribed in mouse forelimbs, while the gene encoding the closely related transcription factor TBX4 is expressed in hind limbs. Could these two transcription factors be involved in directing forelimb versus hind limb specificity? The loss function data were equivocal. Humans heterozygous for the TBX5 gene have Holt-Oram syndrome, characterized by abnormalities of the heart and upper limbs. The legs are not affected, but neither are the arms transformed into a pair of legs. 1998 and 1999. However, several laboratories provided gain-of-function evidence that TBX4 and TBX5 specify hind limbs and forelimbs, respectively. First, if GF-secreting beads were used to induce an ectopic limb between the chick hind limb and forelimb buds, the type of limb produced was determined by the TBX protein expressed. Those buds induced by placing FGF beads close to the hind limb opposite somite 25 expressed TBX4 and became hind limbs. The buds induced close to the forelimb opposite somite 17 expressed TBX5 and developed as forelimbs wings. Those buds induced in the center of the flank tissue expressed TBX5 in the anterior portion of the limb and TBX4 in the posterior portion of the limb. These limbs develop as chimeric structures, with the anterior resembling a forelimb and the posterior resembling a hind limb. A chick embryo was made to express TBX4 throughout the flank tissue. By infecting the tissue with a virus that expressed TBX4, limbs induced in the anterior region of the flank often became legs instead of wings. Thus, TBX4 and TBX5 appear to be critical in instructing the limbs to become hind limbs and forelimbs, respectively. Induction of the apical ectodermal ridge. As mesenchyme cells enter the limb region, they secrete factors that induce the overlying ectoderm to form a structure called the apical ectodermal ridge (AIR). This ridge runs along the distal margin of the limb bud and will become a major signaling center for the developing limb. Its roles include 1. Maintaining time beneath it in a plastic, proliferating phase that enables the linear proximal distal growth of the limb. 2. Maintaining the expression of those molecules that generate the anterior-posterior thumb-pinky axis. And 3. Interacting with the protein specifying the anterior-posterior and dorsal-ventral axes so that each cell is given instructions on how to differentiate. Factor secreted by the mesenchyme cells to induce the AIR is probably FGF10. FGFs, such as FGF2, FGF4, and FGF8, will also induce an AIR to form, but FGF10 appears to be the FGF synthesized at the appropriate time and in the appropriate places. FGF10 is capable of inducing the AIR in the competent ectoderm between the dorsal and ventral sides of the embryo. This junction is important. In which a limb bud is dorsalized and there is no dorsal ventral junction, as in the chick mutant limbless, the AIR fails to form, and limb development ceases. Generating the proximal distal axis of the limb. The apical ectodermal ridge, the ectodermal component. The proximal distal growth and differentiation of the limb bud is made possible by a series of interactions between the limb bud mesenchyme and the AIR. These interactions were demonstrated by the results of several experiments on chick embryos. 1. If AIR is removed at any time during limb development, further development of distal limb skeletal elements ceases. 2. If an extra AIR is grafted onto an existing limb bud, supernumerary structures are formed, usually toward the distal end of the limb. Institute for Advanced Studies, Jodhpur 39. 3. If mesenchyme is placed directly beneath the wing AIR, distal hind limb structures toes develop at the end of the limb. However, if this mesenchyme is placed farther from the AIR, the hind limb mesenchyme becomes integrated into wing structures. 4. If limb mesenchyme is replaced by non-limb mesenchyme beneath the AIR regresses and limb development ceases. Thus, although the mesenchyme cells induce and sustain the AIR and determine the type of limb to be formed, the AIR is responsible for the sustained outgrowth and development of the limb. The AIR keeps the mesenchyme cells directly beneath it in a state of meiotic proliferation and prevents them from forming cartilage. Workers 1989 found that if they cut away a small portion of the AIR in a region that would normally fall between the digits of the chick leg, an extra digit emerged at that place. The progress zone, the mesodermal component. The proximal distal axis is defined only after the induction of the apical ectodermal ridge by the underlying mesoderm. The limb bud elongates by means of the proliferation of the mesenchyme cells underneath the AIR. This region of cell division is called the progress zone, and it extends about 200 micrometers in from the AIR. Molecules from the AIR are thought to keep the progress zone mesenchyme cells dividing, and it is now thought that FGFs are the molecules responsible. When the AIR is removed from an early limb bud, only the most proximal parts of the stylopod are made. However, if an FGF-containing bead is placed in the hole left by the removal of the AIR, a normal limb will form. When the mesenchyme cells leave the progress zone, they differentiate in a regionally specific manner. Cells leaving the progress zone form proximal stylopod structures. Those cells that have undergone numerous divisions in the progress zone become the more distal structures. Therefore, if the AIR is removed from an early stage wing bud, the cells of the progress zone stop dividing, and only a humerus forms. If the AIR is removed slightly later, humerus, radius, and ulna form. Proximal distal polarity resides in the mesodermal compartment of the limb. Information somehow instructing the indifferentiated mesoderm beneath it as to what structures to make. Then older AERs combined with younger mesoderm should produce limbs with deletions in the middle, while younger AERs combined with older mesoderm should produce duplications of structures. This was not found to be the case. Rather, normal limbs form in both experiments. But when the entire progress zone, including both the mesoderm and AIR, from an early embryo is placed on the limb bud of a later stage embryo, new proximal structures are produced beyond those already present. Conversely, when old progress zones are added to young limb buds, distal structures immediately develop, so that digits are seen to emerge from the humerus without the intervening ulna and radius. The meiotic state of the progress zone is maintained by interactions between the FGF proteins of the progress zone and of the AIR. FGF10 secretion by the mesenchyme cells induces the AIR, and it also induces the AIR to express FGF8. The FGF8 secreted by the AIR reciprocates by maintaining the meiotic activity of the progress zone mesenchyme cells. Hox genes in the specification of the proximal distal axis. The type of structure formed along the proximal distal axis is specified by the Hox genes. The products of the Hox genes have already played a role in specifying the place where the limbs will form. Now they will play a second role in specifying whether a particular mesenchymal cell will become stylopod, zygopod, or autopod. The 5 feet ABDB-like portions paralogs 913. Institute for Advanced Studies, Jodhpur 40. Of the HOX-A and HOX-D complexes appear to be active in the forelimb buds of mice. The expression patterns of these genes, and unnaturally occurring in gene knockout mutations, Davis and colleagues 1995 propose a model wherein these HOX genes specify the identity of a limb region. For instance, when they knocked out all 4 loci for the paralogous genes HOX11 and HOXD11, the resulting mice lacked the ulna and radius of their forelimbs. Similarly, knocking out all 4 HOXA13 and HOXD13 loci resulted in loss of the autopod. Human tests for HOXD13 mutation show abnormalities of the hands and feet wherein the digits fuse, and human patients with homozygous mutant alleles of HOXA13 also have deformities of their autopods. In both mice and humans, the autopod, the most distal portion of the limb, is affected by the loss of function of the most 5 feet HOX genes. The mechanism by which HOX genes could specify the proximal distal axis is not yet understood, but one clue comes from the analysis of chicken HOXA13. Ectopic expression of this gene, which is usually expressed in the distal ends of developing chick limbs, appears to make the cells expressing it stickier. This, in turn, would cause the cartilaginous nodules to condense in specific ways. As the limb grows outward, the pattern of Hox gene expression changes. When the stylopod is forming, HOXD9 and HOXD10 are expressed in the progress zone mesenchyme. When the zygopod bones are being formed, the pattern shifts remarkably, displaying a nested sequence of Hox gene expression. The posterior region expresses all the Hox genes from HOXD9 to HOXD13, while only HOXD9 is expressed anteriorly. In the third phase of limb development, when the autopod is forming, there is a further redeployment of Hox gene product. HOXD9 is no longer expressed. Rather, HOXA13 is expressed in the anterior tip of a limb button in a band marking the boundary of the autopod. HOXD13 products join those of HOXA13 in the anterior region of the limb bud, while HOXA12, HOXA11, and HOXD1012 are expressed throughout the posterior two-thirds of the limb bud.